this Friday, you know what we do. We have a fantastic speaker, a friend for many years, and a person who clearly is interested in life. And that may be a best summary of José. He has contributed so many things on many different levels, science and administration and organizing all of us. And it's just phenomenal that he could join us today. Wanted to remind you again about those two initiatives and waiting for your nominations. We will organize those seminars later during the year. So if you have somebody outstanding in your group, your colleague that you wanted to recommend, please contact us and we will go from there. All right, this is all what I wanted to say. Let's just move to Chris Sander.
Hello, everyone. My name is Chris Sander, and I would like to welcome you all to this week's virtual ophthalmology research seminar hosted by the UCI Center for Translational Vision Research. Today, we have the distinct honor of hosting the Distinguished Professor and Chairman of the Department of Ophthalmology and Director of the UPMC Eye Center at the University of Pittsburgh, Dr. José Sahel. Dr. Sahel took his medical degree from Paris University Medical School, followed by residency and fellowship at Louis Pasteur University in Strasbourg, France. He completed a research fellowship at Harvard University and then became a professor of ophthalmology, first at Strasbourg University and then the Sorbonne Université in 2002, where he remains a professor of ophthalmology. Dr. Sahel became the professor and endowed chairman of the Department of Ophthalmology at the University of Pittsburgh in 2016, and has expanded his department to over 20 principal investigators since joining. Dr. Sahel has received a tremendous number of awards, distinctions, and honors, from a thesis medal at the start of his career to the position of officer in the French National Order of the Legion of Honor. Among other highly enviable discoveries in gene and stem cell therapy, Dr. Sahel was the first to hypothesize and demonstrate a rod-derived signaling protein responsible for rescuing cone cells. He and Doctors Monhand Said and Leviard identified the underlying signaling molecule as the rod-derived cone viability factor, which they found stimulates aerobic glycolysis and acts as an antioxidant. Dr. Sahel has founded several companies involved with clinical trials to apply his work to create treatments for patients with various forms of retinal degeneration. We're all very thrilled to have you here today, Dr. Sahel, and thank you very much for joining us.
Well, thank you very much. It's really an honor and a pleasure also, because seeing a lot of friends, and thanks for the ones that are going to bear with my repeated talk. So this is the topic that Chris selected. I had offered an alternative topic to make it a bit more original, but I'm going to show you historical data, but also more recent data. And if you stay up to the end, I will also show very recent data that are in press now, but very promising data. So I've been working for the past many years on trying to protect and potentially rescue cone function in retinal degenerations. So this is not actually my work, it's the work of a very large team of people. You named two important players, but I'll try not to forget too many of them. And this has led us to hypothesize and demonstrate a key mechanism and try to translate that in the clinic. And over the past 10 years, we have also attempted to restore some visual function. And this is based on using this technology called Optogenetics. So this will be the second part of my talk. And at the end of it, I will refer to some thinking about the design of clinical trials and outcome measures in many of these trials, if you stand this talk until the end. So just my disclosures, as you mentioned, I've formed many companies, but the ones that are relevant for this talk today are GenSight Biologic for the Optogenetics trial and Sparing Vision for the development of Rbcvf. I got funding from Novartis many years ago when we started the work on Rbcvf, and I've always had a strong support and commitment to Foundation Fighting Blindness. So I don't think I have to remind every one of you that we rely heavily on the cones, although they represent the minority of the cells in the retina. And this is not only in day vision, but even in a pretty dark environment at moonlight, we are relying on cone function. And obviously, this is also very important for color vision all across the spectrum. The cones are forming the backbone of the circuitry in the retina. As you know, the thinking is that the rod circuitry, at least in part, is piggybagging on the cone circuitry. So they represent the architecture. And when you try to figure out how this works, it's really a mosaic of cone-driven responses with on and off responses deriving from that all across the retina. And it's possible to dissect a lot of it. And this is very important, obviously, for trying to develop new therapies. Cones are also very important for central vision. We use the picture of this painting by Renoir of Mrs. Henriot, where the face is crisp and quite crisp and sharp, whereas the rest of the body is kind of fuzzy, like our peripheral vision, although obviously the gaze is wandering all around to different parts of her. But this is what is functioning in vision, but we are relying on this very small number of cones for high resolution vision. So for all these reasons, cones are really at the core of a lot of research. And I think we were among the many groups working on that over the past 30 years. Loss of photoreceptors occurs in many diseases, and the most important is certainly age-related macular degeneration leading to loss of central vision, like Stargardt disease. But this is certainly a secondary mechanism, the loss of photoreceptors. At least it may not be a primary phenomenon. In retinitis pigmentosa, this is more obvious that the cone photoreceptors and rod photoreceptors are degenerating early stage of the disease. So when we tried with Boton Rosca, my old friend, to review what is done currently, we found some sort of paradoxical situation, which is that the more the disease is progressing through different steps, the loss of rods and eventually of cones, the bigger is the therapy that you need to apply. And actually the outcomes don't go with the size, they get smaller, which means that when you start to try at the best, you would ideally have a small molecule or maybe a vector, which is a bit bigger. And this is where John Flannery and others have pioneered so much work. When you go to cell therapy, it becomes even bigger. And I know that Baruch is involved in many of these. And when you get to implants, and with my good friend Daniel Palanker and also with Mark Humayun, we worked a lot on that, you get to very big systems that actually don't provide that greater vision, but still are able to provide some useful vision. And even better now with new technologies. So when you look at the spectrum of a disease, there is this sequential loss of rods and cones that can be figured as we tried in this with Denis Dalcara in another review. And this loss is occurring initially not only at the level of rod photoreceptors, but more importantly, at the level of the outer segment of photoreceptors. And it's only at the very late stages of the disease that there is a total loss of both cell bodies and outer segments, which provides opportunities for various therapies. At the very early stages of the disease, gene correction or gene editing could potentially be the cure for the disease because it was going to fix the gene defect. But also, it requires that photoreceptors are still alive and viable. Trophic factor could start functioning from the early stages up to a stage where you still have cone photoreceptors, but potentially have enough segment to regrow and maintain the function. And we have been working on this idea of trying to protect them at this stage of the disease. So back to corrective gene therapy, this is now something which has become very popular, especially with the approval of Luxturna, and many of the trials are ongoing and a lot of promises. But there are a few limitations to corrective gene therapy. First one is the number of gene defects. But some gene defects are unknown, that recessive genes and loss of function are probably easier to target as long as they fit into a vector. But there is also this issue that many patients don't come to us at the early stages of the disease, and they already lost a significant number of photoreceptors. And even if they still have photoreceptors, as was demonstrated by Art Silesian and Sam Jacobson, potentially, you could have a continued loss of photoreceptors even after correction. Still, this is the ideal therapy, and this is certainly the goal, to really cure the disease by correcting the gene defect, especially the loss of function. But many years ago, when the first gene defect was identified, I was puzzled by the course of the disease. And this is just a sketch you find on the site of a Moroni Center. The evolution of the disease is that you have a sequential loss of rods and cone photoreceptors. And this had been observed many years ago by Matt Laverne and many others, and demonstrating that this sequential loss is actually also occurring at the level of photoreceptors, and then is driving a lot of changes in remodeling in the inner retina, inner layers of a retina. This was quantified, and you can observe that there is an initial loss of rod photoreceptors. This is the RD1 mouse that Cathy Bowes found the gene defect with Deborah Farber years ago. And then you have this loss of cones, which is more or less mutation independent. And then you have this degeneration of the inner retina. But there is a window where the rods have died, and the cones, although they may have lost already part of the outer segment, are still there and are still very important because our patients are really raving about keeping this function for as many years as possible. So when you look at the disease, it's really a rod cone degeneration, as the name is telling us, and it's really starting at the periphery with rod loss, and then eventually peripheral cone loss and central cone loss. So many years ago, when the first gene defects were identified, it was rhodopsin and then the phosphodiesterase. Most of the initial genes were expressed in rod photoreceptors. Now we know many more genes that are expressed in both cell types, but I found puzzling that the cones die when they don't have a gene defect that explains that. And the idea is that if you can protect the rods, the cones, you might be able to protect very useful vision. Paul Sieving, many years ago, working on Stargardt disease using psychophysics studies, showed that you can lose up to 50% of your cones and still have 20/20 vision. You can lose almost all cones except for 5% and not be fully blind. And Alan Wright, comparing the retinitis pigmentosa to congenital stationary eye blindness, noticed that patients with CSNB don't complain of med symptoms, most of them, some of them do. Whereas if you could transform RP, which is a progressive blinding disease, into a stationary disease like CSNB, you would help a lot of people in the world. So this was at the time we were thinking about it. And I remember that I put on the paper a list of hypotheses. At that time, none of them was explored, from toxic hypotheses, from loss of structure, vascular changes, changes in connectivity, changes in glial responses, especially microglia, oxidative stress, cone starvation, but at that time I didn't know about, and the trophic interdependence of photoreceptors. So we started with a toxic hypothesis, and we published on that many years ago. And we also started in parallel to look at the trophic interdependence. And this idea came from the fact that the loss of cells doesn't occur just in the area of the mutation, but it looks that there is some correlation between the loss of photoreceptors, rod photoreceptors, and cones. For example, in 1998, I think Artsy Desian published a paper in PNAS on postmortem studies in patients with rhodopsin mutation and showed that actually there was some sort of correlation between the loss of rod outer segments, if I remember correctly, loss of more than 75% of them, and loss of cones. And this was in regional variations of the expression of the disease that occurs especially in rhodopsin mutations. So this idea of trophic independence has also been hinted at by people working in the various models like chimeras, RDS, Tom Ray also in vitro. But the idea is that in that case, it's not just rods talking to rods or cones talking to rods, it's really rods talking to cones. And we started the work with a transplantation approach. At that time, with Sadek Mohamed, we were trying to replace photoreceptors, and we used the technology that Martin Silverman, the late Martin Silverman, had developed, which is using vibratome to section pure layers of photoreceptors and to apply that to transplant that in a mutant retina. We use the RD1 mouse at a stage where all rods have degenerated and try to observe rescue of cone photoreceptors. And what we found out with this approach, that there was a rescue all across the retina, not just in the area of a transplant, and it was more than 50% rescue as compared to no surgery, inner retina transplantation, mutant retina transplantation, and MOG surgery. Because the effect was diffuse, with David Hicks, we developed this co-culture model where we observed that actually, as long as you have a retina that contains rod photoreceptors, you have a rescue effect on cone photoreceptors, and this corresponded to 50% of the loss of photoreceptors. I won't get into all these old papers because this is just what led us to identify the factor. Thierry Leveillard joined me at that time for Pierre Chambour's laboratory, and we developed this expression cloning technology strategy to screen for all the genes expressed in the retina on a functional model, which is the cheek cone embryo retina that had been developed by the late Ruben Adler, but by default is mostly a cone pathway, and you have a lot of cones, more than 60, 70%. And we screened 200,000 genes by pools of 110, and eventually this led us to identify the trophic factor released by rod photoreceptors that is supporting cone photoreceptors. This gene was unknown at that time, and we found out in silico that the gene is actually encoding for two different proteins. There is an intron retention. The short form of the gene is the diffusible factor that we called rod-derived cone variability factor, corresponding to one exon. Whereas if you have the two forms of the two exons, you have a protein that is longer and has a function as a thyroidoxine. So the gene is encoding for two different functions. One is really acting as a cellular communication protein, and one is acting inside the cell in a cell autonomous manner. So you have a cell non-autonomous function and a cell autonomous manner. The gene was called nucleoredoxin-like protein number 1. It's a new family of gene, and we cloned also the second member of the family. We found that in the RD1 mouse, the gene was able to rescue, at that time we used a protein, and we were able to rescue part of the degeneration. In the RD1 mouse, it's very difficult to get any function. So we used also the P23H rat that Matt Laverde had developed. And we found in the P23H rat that we had a very significant rescue in functionality. And you see a quite big standard deviation, but it's still significant. It's because you are developing subretinal injections. And this was using protein injection at that time. And when we looked at the number of cells, we found that there is an increased density of cells as compared to the control. But the most striking feature was that in the treated retina, the photoreceptors had outer segments, whereas in the untreated retina, there was only the inner segment or shortening. This is flat mounts from the in vitro, from the treated retinas. And this corresponded to very significant rescue. Then John Flannery proposed that his lab would use new vectors that were developing to try to rescue in the RD10 mouse and actually another model. And they found a beautiful rescue using both the short and the long form of the gene, better with the long form of the gene. And if you look at the density of cone photoreceptors in the treated versus untreated, this is very significant. Importantly, there was also a functional measurement. The ERG was improved, so it's Leah Byrne and Denise Dalcara in the John's laboratory. This was also correlating with an improved density of both the LM and the S cones in the treated retina. They also used an intravitreal administration, and they found a similar strong effect of a rescue with the short and the long form of the gene, but the two of them being administered independently. Thierry in the lab was working on trying to understand the function of Rbcvf. So one of them was to try to understand the function of a long form of Rbcvf. Because there was a strong homology between the long form and the thyroidoxine, the idea was to look at what happens in the mutant, the knockout mouse. We generated the knockout mouse for Rbcvf on the NXLN1. And what was observed is that in the retina of a knockout mouse, there is a strong accumulation of malonaldehyde and markers of oxidative stress. This was not occurring in the brain in these mutant mice, so this was very specific to the retina, and Rbcvf1 is expressed only in the retina. And interestingly, when you administer the factor, just the long form of the gene, and you sensitize the model by light damage or oxygen-induced retinopathy, you find out that there is a very strong rescue using Rbcvf-L in this model. And this is a cell autonomous mechanism, which means that the cones are doing that for themselves. The cones are making the long form, they don't make the short form, and the rods are making it for themselves also, and it's a very strong rescue as compared to the untreated. So the hypothesis we had is that Rbcvf is both providing a non-cell autonomous rescue by rod to cone interaction, but also a cell autonomous rescue against oxidative stress. As a reminder, when the disease is progressing, there is an increased exposure of the remaining photoreceptors to light because most of the cells have died. And there is also an increased exposure to the oxygen that is coming from the choroid capillaries because the blood flow from the choroid capillaries and the oxygen consumption in the outer retina is the highest in the body by gram of tissue. So there is everything for photodynamic stress. So no surprise that there is a strong antioxidant and trophic mechanism over there. And when this is lost, this is accelerating the loss of photoreceptors. So Thierry also wanted to understand what is the mechanism of action of a crosstalk between rods and cones. Because it's a diffusible protein, the idea was that there should be a receptor. So it took many years actually to identify the receptor. This was a receptor that had been known already as a receptor without a ligand in the retina called Bassigin-1. And this was identified as the ligand for the short form of Rbcvf. And this was done by many studies. So I'm not going to get into all of these studies showing the binding to the receptor. And also the combination with another very important protein at the surface of cone photoreceptors, GLUT1, a glucose transporter. So the mechanism of action that was deciphered, and the Cell paper is a very detailed paper on all of that, is that Rbcvf is binding to Bassigin-1, is released by rod photoreceptors, binding to Bassigin-1, and potentiating the function of GLUT1, that is enabling the entrance of glucose coming from the choroid capillaries into photoreceptors. Interestingly, once the glucose gets into photoreceptors, it's metabolized through a very specific pathway called aerobic glycolysis. So it's not the classical glycolysis, nor the glucose, the pentose phosphate pathway. It's a specific pathway that is leading to making lactate, but making also carbohydrates that are very important for the buildup of the outer segment. This is called the Warburg effect, which was described by Otto Warburg in the 30s. And Warburg discovered that actually in 24, he got the Nobel Prize for that. He found that in cancer cells. And Warburg was stating that this was a very specific mechanism to make membranes, but he found that there was no normal cell in the body that had this specific metabolic pathway. Only one exception, that was the retina, and especially the avian retina. You have all the species. And he said it's probably an artifact. And this is a review by Krebs, another Nobel laureate, about that. And the hypothesis was that the fact that you find in the retina this Warburg effect was an artifact caused by tissue damage. It's not an artifact, it's a key mechanism for the maintenance of central vision in patients. So when we published the paper, Connie Sepko is a competitor, more or less, but she commented on our paper. As you know, she has been working on cone starvation, and she hypothesized that glucose was important in cone survival. And actually, Rbcvf is putting the story together very nicely because it's enabling the entrance of glucose into the cells. And the schematic is that when Rbcvf is disappearing, the cones don't have the ability to get glucose inside and to use this glucose through the aerobic glycolysis to make up the membrane. So the outer segments are shrinkening, and this is leading to the loss of cone photoreceptors. Whereas if you bring back Rbcvf, you could potentially reactivate this mechanism. So it was nice to see this news and views. It does fit very well with what we had observed in the P23H rat, but in the treated animals versus the untreated, there is a regrowth of the outer segment, or at least a maintenance of the outer segment in the treated animals. And Thierry put together a schematic saying that rods and cones are married for life. But as when he gave a talk once in San Francisco, I got this comment from Eugene de Juan that life is more complicated, married life, and that's true. Rods talk to cones, but this is a very complex story with both the long form and the short form, the antioxidant activity and the metabolic activity. What is important is that we need both. We need the short form for the trophic effect, and we need the long form for the antioxidant effect, and the two are synergistic. So we came up with this idea that because we cannot recapitulate the alternative splicing that occurs in physiology, we had to administer the two factors. The work by John Flannery's group had shown that each independently had an effect, and we thought that bringing the two together would have a synergistic effect, as the biology I was trying to illustrate in the previous slide was telling us. And indeed, when you put the two of them together, and we tried that in the RD10 mouse, as compared to what you see on this optogenetic test, where the animal is losing vision at P50 as compared to the normal animal, in the treated animals with a combination of the two, the short and the long form in the same vector, because Rbcvf is a very short, NXNL1 is a very short gene, you can put all the two of them together. And we had a very good collaboration with Jean Bennett to start with to do that. What we showed is that the two together are really producing a very good combination and a very good protection as compared to what happens in all the controls untreated, treated with sham or with GFP. And if you project that into what happens in terms of loss of vision, the optokinetic test in the mouse is like a surrogate of visual acuity. This could correspond to decades of vision protection, but obviously we have to demonstrate that in patients. So this brought us to this path of trying to translate that into the clinic. And one of the very important questions is, does it work in a large animal model? And is it safe? So John Fladry had put us in contact with Maureen McCall in Louisville that has this beautiful model of a transgenic pig with rhodopsin mutation. So we started to collaborate on that. And actually we had done work initially with the vectors that John had developed with a short form of a gene, but we put the two together. And as you can see on this preliminary figure, I think Maureen is going to present, Jennifer from her lab is going to present at ARVO very soon, more data, more quantitative. If you compare to the control, the treated animals with the two genes have a much larger number of cone photoreceptors and much nicer outer segments. Interestingly, also, it looks that the synaptic region looks better. This is qualitative data. It's not quantitative data because the animals are a bit different, but this is going in the right direction. And in parallel, the Sparing Vision, the company that was formed to develop a product, has conducted tox studies in the primates, but so far are looking good. So the objective of the studies that we are now preparing, the plan would be to be in the clinic in the next year or less, is to transform a progressive disease into a stationary disease, but not to rescue a vision that is lost, but at least to protect central vision and light-adapted vision in our patients. There is an evolution of the disease that leads over time to the loss not only of the outer segment of cone photoreceptors, but maybe also to anything that could lead to regeneration. Interestingly, in post-mortem human retinas, as was shown in this paper, and Anne Milam had published a lot of data on post-mortem studies in human eyes, showing that there is a stage of a disease where the patients are blind, but you still have remaining cone photoreceptors. And Matt Lavelle sent to Boton Rosca and I a stack of 13 different papers in many animal models, showing that this is a pattern that you find in many animal models. So Boton Rosca, in his laboratory, started with this idea that maybe you could try to reactivate these dormant cones. So this would be a segue to rescuing the cone photoreceptors, would be how to reactivate that. So we started a collaboration on that many years ago, 12 years ago, that led to this idea that maybe using optogenetics, you might be able to rescue the remaining cone photoreceptors, or as you will see, other cells in the retina. So this is at the stage where you don't have outer segments, or you may not even have cells remaining in the retina. So we applying this technology called optogenetics that is replacing the very complex machinery that we have with phototransduction with so many proteins and this beautiful amplification of signal that occurs in the retina, in vertebrates, with a very elementary system that exists in algae that is coupling in the same protein the absorption of light and the activation of a channel or a pump through a membrane that is leading to an electrical signal. So this is a very rudimentary evolutionary process, but it's a very robust process. It comes at the expense of accuracy and the ability to adapt to different levels of light. So the idea was to activate the remaining cells in the retina using this type of opsin, which is microbial. Other groups, including John Flannery, are working on using a more phototransition-like system, maybe not recapitulating all of that, but just bypassing some of it, but using some of the existing phototransduction, which provides probably the ability to function at lower levels of light and with more amplification of the signal. But this is what we started to focus on, to try to replace the activation of cone photoreceptors by light, expressing a pump, which is a halorhodopsin, a chloride pump that is coupled to a chromophore. So you have really, in optogenetics, the idea would be either to activate the remaining cone photoreceptors or the inner retina, could be bipolar cells, could be ganglion cells, and many of these approaches are currently being developed by many groups, but I'll talk about our own work on that. I'm biased to talk about my work versus the work of others, but clearly there is a lot of very good work in this field. So when we started this work with Boton, what was shown is that in the blind mice, the reactivation of dormant cones was inducing a nice spectrum of responses to light, This is hair rhodopsin, but also behavioral responses that the mice were able to function in this optokinetic testing. It was possible to record the ganglion cells and to show that there is a good spectrum of responses. So you can record from cone photoreceptors or from ganglion cells. So we were very excited with this work, and we got funding from Foundation Fighting Blindness to translate that into clinical studies. The good reason for that is when we looked at our database of patients with advanced retinitis pigmentosa, we found that a couple of patients, probably around 20% of patients, still have remaining cone cell bodies, although they are blind, and they still have these cone cell bodies that could be reactivated. So we started this study. Sorry, but there was some frustration, was that the protein was not getting very well to the membrane. And actually, because in primates, we had to do with normal primates, there was some competition with the normal rhodopsin. And so Denise Dalcara, who took over the project, was really trying to optimize all of that. And to optimize the gene therapy, as she learned very beautifully with John, there are different ways to do that, to work on the application route, the capsid, the promoter, and the dose of the vector. So she worked on all of these aspects and screened in many primates what we could do. We have a platform for primate research where we can apply all the surgical tools and everything, and screening from different vectors and looking at the level of expression, and also post-mortem human retinas, and also currently on organoids. She continued the work that they started with Leah Byrne in John Flannery's lab that led to the identification of a 7M8 variant that is very strong in terms of transfecting many cells in the retina. And this led to trying to optimize that, and she found out that the AAV9 7M8 was quite efficient as compared to the native AAV9 to really transfect quite nicely the retina and have a very strong transfection. But on top of that, there were many things to optimize. One was the promoter, and clearly there is one promoter that works very well on cone, which is the truncated natural promoter that the group in Florida had identified, and that is being used actually for different ways to transfect cone photoreceptors. But we also wanted to have a better protein as compared to the protein we were using, which is the hair rhodopsin. At that time, Ed Boyden at the MIT contacted me, and he told me that he had cloned actually 2 new proteins. One was JOES, which is also a hyperpolarizing protein, and another one was Crimson R, which is a depolarizing protein. So we tried to couple all of that, and this is work that Denise conducted, showing very beautiful transfection of cone photoreceptors in the primate macula, and trying to optimize that. And using this approach that can be applied either subretinally or intravitreally, because 7M8 can work both ways. It was possible to record from single cells. So this was work conducted by Serge Picot, Jens Dubel here at the Vision Institute. And you get a very good light intensity curve of responses and a very good transfection of cone photoreceptors. Not all of them, but very significant. What Denise tried to show is that you can use either the intravitreal route or the subretinal. The issue with subretinal is that the detachment of the macula can potentially induce some damage. So the idea was to inject outside the macula and get the bleb to get under the retina. And what you get is a very good transfection of cone photoreceptors. And the titer of vector you need for that is around 10-fold the titer you need for the intravitreal route. This is the type of response you get with subretinal. So the 2 slides can compare, but you get 5 times better response with twice lower dose with this route of administration. So Denise has now optimized that, and she has shifted from JOES to another molecule, which is very promising, but it's her work, and she's going to report on that very soon. And this is an approach that is currently underway to really develop an approach to transfect the cones, the remaining cones in the retina, and using some of the existing transduction or bypassing it. And this would correspond, this is a database, we looked at a sample of patients that we had we follow in Paris. We have a very large database in patients, and showing that there is a good proportion of patients that have remaining cones. But you still have a large number of patients that don't have remaining cones. So what do we do with that? First idea is, how do you replace the cones? And the problem is that if you try to transplant cones, and you can make cones, how do you transplant them? They have to regrow the outer segment, which means that they have to re-engineer all the outer segment machinery. They also have to get in good contact with the RPE to maintain the outer segment and all of that, but they also need to connect to the downstream neurons. So is this going to work? The idea was to deal with this part of the issue, which is really how do you regrow the segment, how do they connect? And to do that, the idea was to use optogenetics, so combining cell therapy and optogenetics. So what has been done, and this was done by the team with Denise and Olivier Gouraud here, was to develop proteins that would be transfected in cone photoreceptors, and I'm going to show you the idea. You take iPS cells from donors, creating organoids that contain a large proportion of cones, transfecting them with an optogene, doing a sorting of the cells to isolate the cones, and injecting these cones into the subretinal space in blind mice. So that sounds kind of crazy, but this was done, and we published that already a couple of years ago. What you get is these organoids. You get a significant number of cones, and they are recordable. The activation with optogenetics works in these cones, and you see pretty good responses that you can observe. This is a good proportion of the cones you get into that, and they can survive quite long, and you get quite good optogenetic responses in that. So this was injected in the animals, and it was possible to record from the transplant itself, and it was also possible to record from the ganglion cells, showing that there is most likely a good connectivity. And then there was some sort of, so it was done by multi-electrode recording, patch rank recording at different levels, and showing that you can get on and off responses at the level of the ganglion cells using a multi-electrode recording. And you can do patch clamp recording, or you can also do holographic measures, biphoton microscopy measurements at the level of cone photoreceptors. And there is some level of behavioral response. And we don't make too much of a story on that because this is a very crude behavioral response you get in these animals. So this study is now leading to trying to develop this approach as a potential therapy. It's a bit complex, but this would correspond to patients that have no cones left and where you would try still to build the on and the off responses in this patient. It's far from being in the clinic. In parallel, the obvious route is to go to ganglion cells. So as you know, the first to have done that was Pan many years ago, published in Neuron in 2006, using the first channel rhodopsin that needs a lot of light to be activated. This led to the trial by Retrosense, which we don't know exactly the outcomes. It was already started a couple of many years ago. The good news, this is work done by Sam and Arti Desian, showing that you still have remaining ganglion cells, even in the advanced stages of the disease. So you could potentially transfect the ganglion cells. The issue with ganglion cells is that you don't select the on, the off, or the on-off, you transfect all of them, but potentially this could work. Because we wanted to optimize that, we first worked on an optimized channel rhodopsin with Hans Bamberg called CATCH, that is still functioning in the green or blue light, and we got pretty good responses. This is the work that Bai and Pan had published in Neuron years ago, so this is very well-known work, but showed pretty good responses. Then we did optimize that using CATCH and could record good responses, including at the cortical level in mice, and did primate studies. But still, this is quite a toxic wavelength. So what we wanted was to get into a wavelength that would be less toxic or not toxic at all. And clearly channel rhodopsin are doing it, but we would like to read really as much as we can into the red spectrum. And this is what happened when Ed Boyden called me and said that he has now developed optimized channel rhodopsin that are shifted into the red. And we had worked initially with another protein that was developed by Chang Nobel laureate, but is a good one too, which is called REACT. But this one was really Crimson R that we decided to try to bring into the clinic. And to optimize that, Denise conducted a lot of work comparing different vectors, different constructs. And eventually what was found is that if you use AV2 7M8 variant coupled with this Crimson R protein anti-D tomato tagging, you get very good transfection and good responses to light in the cells that are transfected. And we try to model the type of visual response you could get. So this is a multi-electrode array in the transfected retina at different levels. And you can really get, and this is published paper that was published in, this is from primate retina, and we also did behavioral studies, was published in Communication Biology a few weeks ago. You get a pattern of responses that does correspond to some potential level of resolution. We tried to model that, not me, but Olivier Marr and Serge Picot, and tried to measure the responses in the retina, knowing that each response to a specific shape has a pattern. And so recognition of letters, you would say so, or shapes by the pattern of response at the ganglion cell level in the retina. And doing that, it was possible to create a mathematical model and to test this model on the measurements, trying to predict the level of visual acuity you could have. And potentially you could reach a threshold of 20 to 2100, which would be great. As you know, with retinal implant, the best of them, the one with Daniel Palanker, you can reach 2500. With zooming, you can reach this type of level of vision. So this is a vision that could be considered as useful vision. Bringing that to the clinic with Untitled Biologics, we had to develop not only the vector part, but also the goggle part. So this was done at the Institute by the team of Serge and the team of Riyad Ben Osman to develop a projector system that is coupled to an event-based camera that is sending the light. The reason for that is that you need a lot of light. Also, although in the red, it's really far below any level of toxicity, but you need also to compensate for the change of levels of lighting because you don't have any adaptation system in the with the optogenetic approach that we are developing. So this was what was developed. So it's a combination therapy with a product, a gene therapy product, administered to the retina, and then stimulation by the goggle. So we just got the paper accepted this week on the first report on the study, so it's going to come out very soon. So I won't disclose that until it's published. It's going to be a few weeks now on the first results of this study that we performed in patients. If you look at the spectrum that we have tried to talk about today, different therapies could be proposed at different stages of the disease, but all of this is really dependent on the status of a retina. How healthy are photoreceptors? How degenerated are them? Are they totally lost? And the type of therapy you can propose will depend on that, which means that the personalized medicine is not just the genotype, it's really the stage of the disease and the status of photoreceptors. Fortunately, we have all these technologies that enable us now to observe photoreceptors using adaptive optics. This is what Ethan Rossi is doing in Pittsburgh, where you can see both the cone photoreceptors, but you can also go from a mosaic and look at the RPE cell. And there are many groups that are doing that currently. Beautiful technology developed by David Williams, Austin Roda. And you can combine the two and reconstruct the density and correlate that with all the measurements we have for the OCT, looking at the easy zone, the length of the outer segment, looking at the autofluorescence pattern, and you can get a multimodal way to try to assess the density of photoreceptors in health and in disease. But at the other end of the spectrum, you want to see what is the benefit for the patient. And for that, visual acuity or visual field may not be enough, and you get into this very blurry, very murky field of patient-reported outcome and performance-based tests. So I'm going to complete my talk on that, just referring to work we started in Paris and now continuing in Pittsburgh, and which is trying to measure visual function in real life. As you know, this is what was part of the approval of Luxturna, and many groups are working on mobility and navigation. But just to give you an idea of what we developed, we tried to develop a metric that could be associated with the functionality. For example, recently a work that was done in the Institute correlated the length of the outer segment in cones during aging with performance in the environment I'm going to show you. We are using a motion capture system on patients where we are monitoring the gait, the direction of the gaze, the mobility of the head, and multi-parameters of visual function that are developed in real life in what we call the naturalistic environment. More recently, we have developed a virtual reality setting to do that in an environment that would be more controlled and more reproducible. And we do that, if you look, for example, at this patient that has this type of visual field, that is moving into the environment, trying to migrate through that, avoiding the obstacles, and finding this door. This is the direction of the gaze. These are the measurements of the gait, of the direction of the head. So you get a lot of metrics, and this should correspond to visual function. So to show you that, this is what you are measuring, the walking cycle. the trajectory, the direction of the head, and the saccades. And as you can see, what is obvious is that the patient with RP is moving the head much more downwards, for example, as compared to a patient that has normal vision. And when you get all these vectors together, looking at saccade amplitude, rotation of the eye, and all of that, you get a pretty good assessment of functionality that is quantitative. And we have now performed a lot of correlation between visual field, for example, or dark adaptation, or contrast sensitivity, and what we see in terms of visual function. So to complete my talk, I just try to describe a spectrum of possibilities from what many people are doing in the field: gene replacement or editing, trophic support, restoration of remaining vision that can be attempted. And I'm not going to talk about all the work we are doing with Danielle on prosthetics, but clearly this is a part of a spectrum of therapies that could be proposed to patients. The good news is that there is hope. The bad news is that there are many holes in the field and still many, many years of work before we can be very optimistic for all our patients, but this is really promising. So as I said, this is the work of many people. I put some of the names on the slide and probably forgot many of them, but and also funding by many sources that was done. And I just complete my talk by showing you the building that is now starting to be off the ground in Pittsburgh, a 10-story building that we are building for the Vision Institute, which will be 450,000 square meters, all integrated around vision and rehabilitation. And that is now, we are at this level of construction, so not yet there, but it's due to be completed at the end of 2022. And obviously, we are just at the start of the battle, but at least there is hope that we can win this fight against blindness from this type of degenerative disease. Thanks for giving me the opportunity to share that work with you. Thank you very much.
Thank you very much, Sahel. Any, Jose, any questions, please drop me a chat that you have one, and we'll go chronologically.
Someone has to start.
Okay, Susie.
Hi. Oh, sorry. Hi, Dr. Sahel. That was a very great talk. I have two questions. First is, in the NXNR1 knockout mice, like at what age do you start to see cone degeneration, and how fast is the progression? And my second question is, have you looked at the expression level of NXNR1 in other retinal disease mouse model, which has a slower raw degeneration and the cone degeneration occur before the raw degeneration?
Yeah, so two great questions. Clearly, on the first one, so we generated the knockout for NXLN1, which is the gene we cloned, and then NLXL2, which is a paralog, which is very similar actually to NXLN1, and both are made by Rhodes and are rescuing cone. So when you knock out one of them, the other one is still there. So there is some redundancy, but you have a loss, spontaneous loss of cone photoreceptors that takes, and actually some rods also are degenerating. But this is like in the range of 20% loss. If you add oxidative stress to that, or light exposure, then you have it's like 50% loss of cone photoreceptors. But again, you have the other one, the Rbcvf2, that is still present in these retinas. When you lose both, when you lose rod photoreceptors, you are losing the two factors at the same time. Then getting to your second question, which was the level of expression. So we looked at the level of expression in different animal models, and I know that this has been done also in dog models. And every time you have a loss of rod photoreceptors, you have a loss of expression of Rbcvf. We also looked at some postmortem retinas, but as you know, these are very advanced patients. So there is a loss of Rbcvf2, but these are very, very advanced retinas. And we also looked at, this is a paper we published in retinal detachment. We got samples of a retina surgically removed during surgery for retinal detachment, and it parallels the expression of rhodopsin, the expression of Rbcvf. So you have a parallel expression in humans. So these are some of the elements that could address some of your questions.
Thank you.
Okay, let's move to Daniel.
Hi, Daniel.
Hello, Jose. Thank you for this very broad review. And It's exciting that your paper is finally accepted to Nature Medicine. I'm looking forward to reading it. Maybe you can comment more about what actually patients report in this optogenetic study. What do they see? How they describe? What kind of tests did you do?
Yeah, so we have done the testing has been mostly mobility and eye-hand coordination. So we, for some of the patients in the study, we are still waiting because of COVID. A couple of things were delayed, but so you'll see the paper hopefully very soon. So there's an ability to detect objects and to quantify objects and to grasp them. So this is in blind patients. So this is what is being reported, and it's associated with cortical activation in the area. So there is we did a multi-electrode EEG to correlate that with object recognition, and so it's a couple of objects, not every object, and eye-hand coordination for that. So, but there will be more details in the paper.
Okay, Kamal.
So it's not reading letters like we have with the implant so far. Yeah.
Hi, this is very nice talk on optogenetics, and I have also. Would you comment, like, last year I think from the ISOCAF lab they had shown using EMW opsin, even the white light, the ambient light, you can still see some of the visual response from the mice. And I believe some of the people in optogenetics, they are trying to combine, like, if you combine celeropsin and crimson R, which has blue, green, and red, and that pretty much gives the wide spectrum kind of white light. Would that be, even possible, combining 3 opsin and having a wide spectrum, and then we can use the ambient light even?
Yeah, I think most of the combinations are trying to use some of the endogenous transduction mechanism and using to have a better opsin. Actually, the paper by Isaacoff was with John Flannery, who is on the talk, and it was published in Nature Communications, I think 2 years ago. But I think there is more to come. So maybe John, given that I think if you are still there, if you didn't leave, maybe you can reply to that question about your data.
I don't want to hijack your talk, Jose.
No, please do. Please do. Please do. John?
The suggestion's good. It's going to be really difficult to put in multiple optogenetic sensors because you're going to need vectors that go to different subsets of the cells. And cell-specific promoters for on and off bipolars or different classes of ganglion cells. So that's a ways off. I don't think it's a good idea to put multiple options with different lambda max in the same cell.
Yeah.
All right, thank you.
Okay, let's move on to Don Zach.
Hi, Jose. That was amazing and comprehensive as always. I have a sort of probably unanswerable question. But with all the different modalities of optogenetics that you and others are doing, neuronal plasticity and how the retina and how the brain interprets it, obviously is going to be a major part of how much we can. So and different people learn differently. What's known about the characteristics that make some people more recipient, having, better ability to integrate these altered signals? Is there any studies going on what kind of patient, what kind of retina is more likely to benefit from these signals than others?
I think there are several parts to your question. One is certainly what is the status of a retina? What is the status of a remaining connectivity? Especially if you talk about reactivating cone photoreceptors, it will depend on the connectivity you have remaining in the inner retina. Same for ganglion cells, really depend on how healthy the ganglion cells that you are stimulating are there. The learning thing is a major thing. The plasticity is a major thing. I mean, when we did the first retinal implant with second sight and stimulating the ganglion cells, clearly it took months to the patients to be able to make sense of the starry sky we were seeing, how to align these planets to make a constellation, and then to start to think, this is a door, this is something. And actually there was some training, even of investigators, on how to help the patient to accelerate this path. With the subretinal prosthesis, it's faster because it's more natural vision, because first of all, the coordination of the eye movements with the object is much better because it's really following the eye, but there is still a learning that has to be developed. For the optogenetic, which are really preliminary, I mean, the report we are publishing is probably one of the first on functionality. It's really the training that took time. It's not immediate. You don't reactivate the retina like you put the bulb and light is there. It takes time to reconnect. And again, we are stimulating ganglion cells. So probably the signal is very confusing to the brain because it's both the on and the off, and the brain has to make sense of that. I guess that eye movements and changes are helping in that respect, but there is certainly something that we are learning down the road on how to provide the best stimulation and how to train the patient to use the restored vision, which is really not what we would call normal vision. This is really some sort of vision that patients have to make sense of.
Thanks.
All right, let's move on to John. John Hong.
Oh, yes. I have a question. Maybe this is, I'm not sure, but for the transplantation of the cones generated from iPSC cells, I was wondering if there's a reason for transplanting the optogenetically engineered cones over just regular cones.
Yes, it's good that because you're fixing something I probably was not very clear about. The reason for that is that when you transplant, let's say that you generate progenitors of cone and you're able to transplant them, they have to regrow the outer segments and they have to reestablish a good connectivity, not connectivity, but a good interaction with the RPE. And it turns out that the transplanted photoreceptors don't always regenerate the outer segment. This is one of the issues, is if you don't have outer segment, they won't function. And if you don't have a good interaction with the RPE, the cycle of vitamin A and everything and the regeneration is not going to occur properly. If you put optogenes into the cells, you don't need any of that. And so this is why this was this idea to overexpress. It came from Jens Stibel initially, and then Denise has continued that. And this is really the idea, is to not to have to rely on the natural transduction, but to try to bypass that.
Right. Thank you. So I guess that sort of like covers many diseases at once if you're able to bypass all of these issues. Well, potentially, yes, but as you can guess, this is a complex technology. So there is a lot of work currently to see how you can accelerate that. iPS cells and organoids take months to be generated. Then you need to do cell sorting, you need to do transfection. So it's a multi-step. I can imagine that the regulatory pathway to get this approved is going to be quite long, but it would certainly address a lot of issues. Yeah. Thank you.
All right, let's move to Emma.
Hi, Dr. Sahel. Good morning. My name is Emma. I'm a Dr. Current postdoc fellow, and I have a very general question. I'm just wondering if every time that you do your studies on photoreceptors, have you looked to what happens to the vasculature, to the retinal vasculature? You know, when they are dormant, does the vasculature degenerate? And then when you reactivate them, does the vasculature recovers or?
Yeah. So we haven't done a lot of that because our thinking initially was that because the photoreceptors are dependent on the choroid capillaris, and the choroid capillaris is less regulated in terms of blood flow as compared to the inner retina. As you know, some level of vasoconstriction in the inner retina as a consequence of a mutation. So we haven't done that too much in the animal models, but the plan we have is, because we believe we'll be able to study that much better in human studies, we have now a very nice way to visualize the blood flow. Something it's not part of my talk today at all, to look at the blood flow using digital holography and Doppler holography to measure the blood flow both at the level of choroid capillaris and at the level of the inner retina. And we are applying that currently to a cohort of RP patients we are following to look at the changes in blood flow in these patients. And the plan would be to see what happens after therapy. But this is a long-term project. We published, I'm just one of the authors on these papers, on these technologies. The main author is Atlan 8 CTLAN. It's published in a lot of optics journals. You'll find a couple of papers on this technology called digital holography, where you can measure the blood flow all across, and you can get into the depth of the retina. You can measure even the optic nerve at different levels. So it's one of the technologies. Others are doing different technologies for measuring blood flow, but this is certainly a very important question. We still believe that this is a secondary event, but an important one.
Thank you.
Okay, Zach.
Hi, that was a great talk. So I was wondering, I had two questions. One's kind of a developmental question. The other is more of a biotechnology question. From a developmental standpoint, is it possible to introduce photoreceptor progenitors that aren't fully matured or developed yet and have them develop in situ so that they establish, a really strong connection with the neurons, for example? Or is the environment already set to really not facilitate that? And then my second question was kind of, can you incorporate small molecules or biological scaffolds to help enhance the seeding of those organoid-derived cones into the retina?
Yeah, so on the first question, actually, this is something that was initiated by Anand Swarup many years ago that led to the work that Robin Ali and McLaren published in Nature in 2006, where they were using the cells at the stage where they enter the differentiation into photoreceptor progenitors. So I think Anand is on the call, so I'm hesitant to talk about his ideas, but so they showed at that time that there was integration and some functionality. As you know, there was an ensuing debate that occurred after where this was a real reconnection or material transfer, and big fights between actually Robin and Rob McLaren on this issue. So I won't get into the politics of that, but certainly this idea has led to demonstrating that at least there is a level of integration and functionality. Still, the interpretation of the data is still not very clear on that, and there is a lot of follow-up work on that, on this idea. So it's a great idea, and maybe, I don't know if Anand wants to comment on that, if he's still with us. I don't know. Anand, are you here? He's not. Okay. And then on the other one, clearly it would be nice to be able to, I think Anand is here. Okay.
Yeah. No, I mean, I'm here, but there's no further comment. As you said, there's a lot more to be done still. So we'll wait for that. Thank you.
There is a lot. It's a great idea. It's Anand's idea actually, and it was a great idea. And the other one clearly, if you maybe I didn't mention that, but initially I thought we could transplant photoreceptors and just use the paracrine effect of a transplant. But we found out it's very difficult to get access to the cells and post-mortem retina and everything. And we had started to work on the scaffold to keep the photoreceptors under that, but this was more a transplantation study or the cell therapy approach. So this is more like the type of approach that Henry Klassen, for example, is developing the paracrine or delivering the trophic factors using a factory, but it's a different type of approach as compared to our idea is really just to administer the factor using gene therapy. So not very innovative, but this is what we would like to do.
Okay, thank you.
Next question will go to Vladimir.
Thank you, Chris, and thank you, Dr. Sahel. A beautiful talk. My question is about the rod-derived cone viability factor. Is there a difference in the dependence of foveal versus peripheral cones on this factor? And when you do the gene therapy also, do you see different degree of protection in the periphery versus the fovea of the treated retinas?
So on the second question, we don't have a lot of clues so far because we have worked on mice and rats, and there is no real good difference between central and peripheral. We know of studies that are ongoing in the pig, and clearly we have the effect is really in the area, especially because we are using a combination of the short and the long form. And in that case, we see the long form is only in the area which is transduced because it's a cell autonomous mechanism. The short form is diffusible. So initially, when Maureen was working with John on injecting the short form in the vitreous, I think they had a diffuse effect on all around the retina. I don't think she did discriminate between the central and periphery. I know that she had done in the past multifocal ERG or pattern ERG, but I don't think she has done that on that model. So I don't have an answer on that. I think this is something we'll see from human studies probably in the future. On the dependence, it looks that there is some sort of regional vicinity, that there is a gradient, and the closer you are to rod photoreceptors, the closer you are to the source. If I'm quoting properly the paper that Art published in 1998, I think in PNAS, in this rhodopsin mutant retinas postmortem, there was a regional parameter. It was in the areas where you had more loss of rods, but you observed also more loss of cones. So there is something like that. Maybe Art, am I distorting your paper, or is it right?
No, you're not distorting it, and thank you for remembering it 25 years later. But the whole concept brings up fascinating questions, at least in my mind, and I will ask a question to you as to what your thoughts are regarding the exceptions to the rule of, cones follow rod depth. And the exceptions that come to mind, at least in my experience, in my more recent interest and experience, are two forms of Leber congenital amaurosis, the CEP290 form and the MPHP5 form, both of which have a precipitous loss of rods and what we think are remaining cones that remain for decades, up to age 40 or 50. So that comes as a very specific example to the opposite, and that brings up the question as to why. And one of the thoughts, at least that comes to my mind, is that maybe it has something to do with the functioning of the remaining cones, that they are more susceptible if they are less functioning. More susceptible if they're more functioning, and less susceptible if they're dysfunctioning. But that idea again brings up another exception, and the other exception, going back to your question about the rhodopsin, is of course there is the rhodopsin version where there is segments of retina, both rods and cones are dying together, but there is the other form of rhodopsin mutations, what we have called the so-called class A, where again, just very similar to the CEP290 and MPHP5, there is a precipitous loss of rods very early in life, and again, retention of cones for a long time, for many decades. So I'll stop the diatribe and ask the question, do you have any thoughts as to how the exceptions may work in terms of the rod-derived cone viability factor?
Yeah, so I think the hypothesis we have on that is that when the cones are deprived with this type of support and they are exposed to more stress, they are turning down the machinery and they are more or less shortening the outer segment as a protective mechanism. Maybe it's maybe not meant to be protective, but it ends up being, they just lose the ability to regrow the outer segment because they lose the support from Rbcvf that is controlling in some ways the growth of the outer segment. So the outer segment is shrinking, and this is a part, it may lead to some sort of survival because they are no longer activated that much by light, and they are just turning down the machinery. And obviously, if you are able to rescue some of that, then they can regrow it, which is what you're observing in some of the studies that you have done. The other exception that I keep hearing all the time is that in some species, they are cone-dominated versus rod-dominated, and the cones are surviving. And it turns out that they are making, in that case, they are making a lot of the autonomous part of the signal. The cones are making a lot of it. So, and again, the cones are making the long form. It's not that we are losing everything when you lose the rods, you lose the buildup of a short form, the expression of a short form by the rods. The long form is made by the cones themselves and by the rod themselves. So it's a combination of the two. Clearly, it would be interesting. The problem is that we don't have good animal models for all of that to study what happens in the macula versus the periphery. But these are some of the tentative explanations I could give to that. Obviously, I love my hypothesis, so I'm trying to defend it. But this is what I believe could be underlying what you described.
All right, we had a wonderful presentation, Joseph. Thank you for that. We have a great conversation. We have one more question, and I'm looking for a victim at that moment. So, Kathy, be sure that you're not asking the final questions. So let's go to Kapil as a final commentator and final questions from him. But before we do that, let's entertain Aisha.
Hi, Dr. Sahel. Very good talk. I have one question. When you did all those transaction with the organ with and cone cells, did you found that there is an immune response from the local immune, from microglia, or from the infiltrated monocyte? And how do you bypass those problems?
Well, actually, the mice are very kind with us. The mice are very tolerant to a lot of stupid things we do. So in the mouse, you don't see a strong immune response in general, especially when you go subretinal. We did not observe much. We haven't looked at all the microglia. This is work which is being done currently, but there was no evidence for a strong inflammatory response to the transplantation. But what you see in the mouse doesn't mean much in terms of immunology. I wouldn't tell that this is not happening if you do that in primates, for example. And certainly this is a key step before you would do that in patients.
Thank you.
Okay, we're going to final comments from Kapil.
Thank you, Chris, for picking on me. I'll say thank you for a wonderful talk. As it's amazing. It's can't even call it tour de force. It's such an amazing amount of work, and it's such an inspiration to hear all this work that you've done. My comment and question is regarding kind of the difference between rods and cones and their metabolism. And obviously, I'll go back to my favorite cell type, which is the RPE. And the little background I want to give is that we're trying to make RP cells that would be macula specific, that would preferentially talk to cones, versus RP cells that would be rod specific, that would be on the periphery side. As you know, they're slightly different in their size and in the apical processes. And we've always wondered, how does this interaction between rods and cones, especially with the Rbcvf, how does that affect the metabolism of RPE? So is there a crosstalk from rod photoreceptors or cones to RPE cells to tell them, hey, we need these metabolites, please send them over. We are the right cell type here. What do you think?
Yeah, well, I think this is work, but the group, actually Maureen has been part of that work. There is a series of work that has been done by the team in Louisville. I think there was a neural paper and a follow-up of a paper where they looked at the transfer of glucose through the RPE to cone photoreceptors. And they showed that there is some regulatory mechanism occurring at the level of the RPE for the delivery of glucose. And just this week, there was a paper from Connie Sepko in eLife, where they looked at lactate transfer through the RPE and the transport. No, they were not exactly. They were looking at cones, but they were postulating that in case the RPE was blocking the transmission of glucose and lactate through the RPE to photoreceptors, they could bypass that. So she's using another gene as well. She's doing similar things, but differently, obviously, competition and cooperation. And clearly, the RPE is certainly very important, at least for glucose transfer, probably more than that, certainly vitamin A we know. So this is part of the story. But what is interesting is that if you look at the effect in vitro of Rbcvf on the cones or on explant and everything, you don't have the RPE and you still have this effect. So it's not dependent on the RPE, but probably there is a synergistic mechanism between what RPE does with glucose transfer and metabolism and lactate and what the photoreceptors are doing. So it's certainly correlated. And there might be, you're right, a regional difference between the central RPE, especially because of the presence of so many cones that they have to talk to in a small area. So it's very likely to be an important synergistic mechanism.
Thank you.
Right. Thank you, everyone. A week from now, we will have Ed Stone. Hope to see you all.